Hello guys, we have completed until here. So we have to complete the remaining syllabus. Let's discuss sensory photobiology today. Not like animals, plants incapable to move from one place to another place like animals. So they have to stay in one place only. So they have to adjust for any unfavorable conditions. Sensory photobiology illustrates the remarkable ability of plants to receive light and respond to the light. The light is in different wavelengths, so plant observes different wavelength photons and respond to the signals. So this light is useful for not only for photosynthesis, it is also useful for its development, seed germination, stomatal movement, also for photoperiodism. So in order to receive that light, they must have some receptors, right? They are phytochromes, cryptochromes and phototropins. So these receptors receive different wavelengths of light and translate these signals into physiological responses. We have completed introduction part. Now let's start today's lesson. Plants possess three major classes of photoreceptors. What are they? Phytochromes, cryptochromes, phototropins. I told you earlier, they observe different wavelengths of light. So phytochromes are observing 660 nanometers. That is called red light. Okay. The another one is 730 nanometers. That is called far red light. The major functions of phytochromes are seed germination, shade avoidance. That means escaping from the shade. How plants escape from the shade? They have to move towards the sunlight. Also, these are helpful in flowering. These are important functions. Please remember these all. Okay. The second photoreceptor is cryptochromes. It observes blue light and UVA light, ultraviolet A light. It is useful in circadian rhythm and stem elongation. What is circadian rhythm? We also have circadian rhythm. Circadian rhythm means it is like internal clock of 24 hours. What things should be happening in 24 hours in our body or in plants. The third one is phototropins. They observe blue light. These are useful in phototropism. What is phototropism? Please comment. I am waiting for your comment. Phototropism means plant grows towards the sunlight like sunflower tree. The second function is stomatal opening. The third function is chloroplast movement. You can see here is red light. Here is far red light. So it is between 600 to 700 nanometers. Basically we say 660 nanometers. The second one is far red light. That is from 700 to 750 nanometers. Here you can see that it observed red light. It is PR. We can see it observed red light. That means it observed between 600 to 700 nanometers light. So this PR, that is in inactive form, converted to PFR, that is in active form. So this activated form stimulates physiological activities in the plants. So what happened here? Red light converted PR to PFR. Here you can see that is from 700 to 750 nanometers light that converted back PFR to PR. So it is converting active form to inactive form. Why? If there is more PFR or if there is no need of PFR, so this active PFR should be inhibited. So it will be inhibited back to PR. So it can change its shape from PR to PFR, PFR to PR. Remember it. These are the functions of light response. What are they? Germination, de-ethylation. Plant needs to absorb light from the sunlight. So plant bend towards the sunlight. Plants stop bending towards the sunlight when it receives sunlight. Okay. So it absorbs the sunlight as quickly as possible. So what happened here? It is not stretching now. It is absorbing sunlight. So we call it as de-ethylation. The third function is inhibition of stem and petiole elongation. This is what I have told you until now. Chloroplast development, chlorophyll biosynthesis, leaf expansion to receive the maximum sunlight, branching, so branches form, flowering, senescence. Senescence means it is like dying. Okay, let us see first receptor, that is phytochromes. The discovery of phytochromes, they were discovered while studying about red light mediated seed germination in lettuce. What is lettuce? It is like a cabbage but not cabbage. These are found during the studying of red light mediated seed germination in lettuce. Okay, observation, red light stimulate germination. So they observed that red light stimulating the germination. far red light reverses the effect. So it reverses back to PR. We have seen it earlier, right? This reversible reaction suggested the presence of photoreversible pigment. Later called this pigment as phytochrome. So let's see about it. Phytochrome is a chromoprotein. Chromo means color. It is a chromoprotein consisting of apoprotein. Apoprotein means we have studied in degree PG. If it is a lipoprotein, that means lipid is here and also protein also present. The protein part we call as apoprotein. So it does not have lipids. So it consists of apoprotein, protein part of phytochrome. Its molecular weight is 120 to 130 kilodaltons. It contains several domains. They are pass domain, gap domain, phi domain. These domains are important for light perception, signal transduction, dimerization. What is dimerization? What is dimer? Did you remember from chemistry? Two monomers forms dimer. Okay. So we call it as dimerization. It also consists of chromophore. Chrome means color, light absorbing pigment. The chromophore is a linear tetrapyrrole called phytochromobilin. So it is in linear form. It is similar to bile pigments in animals. It is like biliverdin, okay. Chromophore is covalently attached to apoprotein through cysteine residue. So remember it. What I can tell you about it? Please remember it, okay. Photoreversible forms of phytochrome. Phytochrome exists in two interconvertible forms. The first one is red light, the second one is far red light. So they are interconvertible forms. PR converting to PFR, PFR converting to PR. PR absorbs red light, that is 660 nanometers. PFR absorbs 730 nanometers, that is far red light. It is inactive form, it is in active form, okay. We have studied earlier. Conversion. Red light converts PR to PFR. Far red light converts PFR to PR. Dark reaction. PFR slowly convert back to PR. Why? Here is no sunlight, so plant cannot observe sunlight. So physiological activity should be stopped. To do that, PFR should be inhibited. PFR should be converted back to PR. So it is helpful in the time of darkness. Phytochrome gene family. In Arabidopsis, there are 5 phytochrome genes. Did you remember what is Arabidopsis? What is it? Is it a plant or animal? Comment it. So it consists of 5 genes. They are phytochrome A, phytochrome B, phytochrome C, D, E. Okay. So we call it as phi A, phi B, phi C, phi D, phi A. Okay. It is an abbreviated form. Functional types. Type 1 phytochromes. These are light labile. They degrade in light. Examples for it is phytochrome A. The type 2 phytochromes are, they are stable in light. They are phytochrome B, C, D, E. So remember it. It is degraded due to light. It is stable in light. Mechanism of phytochrome action. Light perception. Red light converts PR to PFR. We have seen it. The second one is conformational change. The protein undergoes structural change. What is conformations? Do you know? The third one is nuclear translocation. Active phytochrome, that is PFR, moves from cytoplasm to nucleus. Remember this point. So this active PFR moving towards the nucleus. Why it is moving towards the nucleus? Because nucleus synthesizes genes. They activate physiological activities. The fourth point is interaction with transcription factors. PFR interacts with PIFS, that is phytochrome interacting factors. So PFR interacting with PIFS. Examples for it: PIF3, PIF4, PIF5. Phytochrome binding causes PIF degradation via ubiquitination. So what is happening here? PFR inhibiting the functions of PIF. Okay. The fifth point is gene regulation. Activation or repression of light regulated genes. So phytochrome activates and also represses, means inhibits the light regulated genes. So it can function or it can stop.